Welcome back to this next video in which we are discussing HPVs and cervical cancer. Okay, so we're currently in the process of discussing the HPV life cycle, and we've just discussed the role of the E7 protein. So we've seen that E7, via activating the retinoblastoma E2F pathway permanently, is going to cause the continuous activation of the S phase inside these virally infected cells in the mid-zone of the squamous stratified epithelium. And this is going to lead to them dividing very rapidly, and every time they activate the S phase, you're going to get massive viral genome amplification. So the overall result of this is that you get loads of mid-zone cells that are full with viral genomes inside their nucleus. Okay, and this can actually cause a visible growth, which is what we would call a wart. However, it doesn't necessarily produce a visible growth called a wart. What we now want to discuss is the role of the E6 protein, and I've deliberately done E6 after E7 because E6 is really to stop something that might result from E7. So let's just go over here and we'll now discuss the E6 proteins. I'll just put this as a little title. We are now going to discuss the E6 protein. So, I said when we were in the previous video and discussing E7 that what we just said about E7 was applicable for all the human papillomaviruses, all the different types of human papillomavirus, when they infect their respective squamous stratified epithelium, will produce an E7 protein when they're in the mid-zone layer of cells, and this will cause the activation of the retinoblastoma E2F pathway and therefore rapid division. What we're about to say for the E6 protein does not apply for all of the different HPV types. It applies for some HPV types, particularly the high-risk HPV types. So what I'm going to say, it applies for the E6 proteins that are going to be produced by high-risk HPV types. The E6 proteins in the other HPV types that don't follow what I'm about to say, we don't really know what they're doing. So this is a pathway that's really important and really, famous, but it doesn't apply for all of the human papillomaviruses. It's something that the E6 protein of the high-risk human papillomaviruses does. Okay, so this is just the high-risk human papillomaviruses. So, I want to firstly discuss the motivation for what's about to happen. So we've just seen that E7 is going to cause very rapid division in cells, and we also said at the end of the previous video that the E7 protein of high-risk human papillomaviruses is stronger at doing this than the E7 protein of low-risk human papillomaviruses. So particularly in the case of high-risk human papillomaviruses, you're going to get massive cell division. Now, that cell division is not normal cell division. I showed you normal cell division down here. Normally you go through this incredibly ordered series of events, and it is an incredibly coordinated process. Everything occurs at the right stages normally. In normal cell division, it's incredibly coordinated. I mean, you can learn this process in a huge amount of detail. All the different proteins, the cyclin-dependent kinases that are activated at different stages and coordinating this entire thing and activating the retinoblastoma E2F pathway at exactly the right time. And I've just realised that when I copied it, it somehow lost the E there. I'm going to have to rectify that now. So let me just waste a bit of time by putting the E there. Okay, so normally this cell division process is incredibly coordinated and it goes through in this exact order. When the virus, using its E7 protein, is activating this, it activates the S phase first, so you do not have the same normal, coordinated, beautiful process occurring. We could describe the type of cell division that the virus E7 protein is going to be activating as aberrant cell division. Indeed, I will describe it as aberrant cell division. So this is a very key term. Aberrant means wrong. There's something wrong with the cell division process. It's not the normal coordinated cell division process that would occur normally in cells that are dividing because they should be dividing. It's aberrant cell division. Now, there is a pathway inside all of cells called the P53 pathway, and this is going to be absolutely key, so I'll put this as the title, another title. So the P53 pathway, so there's a really important pathway called the P53 pathway, which exists inside all cells, and this becomes activated when you have aberrant cell division. So uncoordinated, unnormal, aberrant cell division activates a signalling pathway called the P53 pathway. And what are the consequences of the P53 pathway? Well, it stops the cell dividing. So, one, it stops cell division. In addition, if it's activated to a high enough extent, it can actually cause the cell to commit apoptosis. So apoptosis can also be a consequence of the activation of the P53 pathway. So this is a pathway that is trying to prevent cancer from forming. It's a pathway that exists inside all cells, and if that cell starts dividing in a strange, stupid way, then this pathway exists there to be activated and to stop the cell dividing, and even if it's activated enough to get the cell to commit suicide for the protection of the meta-organism, the multicellular organism. So this is a really important pathway to protect the multicellular organism. It is the archetypal anti-cancer pathway that all cells have inside them, that is supposed to detect the possibility that the cell is becoming cancerous, and if so, stop that. Of course, it often goes wrong, and that's why cancer can form. And indeed, E6 protein is going to make it go wrong in this case. So there are other things apart from aberrant cell division that can activate the P53 pathway. Also, if you've got DNA damage, this can activate the P53 pathway. So if you've got damaged DNA, then you do not want the cell to be dividing, so it makes sense to stop the cell from dividing, and if the DNA damage is bad enough, for the cell to commit apoptosis. In addition, besides just making the cell stop dividing and committing apoptosis, the P53 pathway also activates the DNA repair mechanisms. So this is another thing to add on here, that it activates the DNA repair mechanisms so that we can repair the DNA that has been damaged. So, overall, there is this incredibly important pathway called the P53 pathway, and we will go through the details of this pathway in just a moment, which is activated by strange cell division and DNA damage, and which as a result causes the cell division process to halt. It puts a brake on the cell division process, it stops it in its tracks. If it's activated to a high enough extent, it causes the cell to commit suicide, and it also activates DNA repair mechanisms to try and fix the DNA damage that might have occurred. So, this spells catastrophe for our human papillomaviruses, particularly the high-risk human papillomaviruses, because they produce a very strong E7 protein. They produce extremely powerful aberrant cell division, extremely wrong cell division, and therefore there is a terrible risk for the human papillomavirus that we're going to get the P53 pathway activated, and therefore we'll get cell division stopped, maybe even suicide of the cell that the human papillomavirus is in, and DNA repair mechanisms activated as well, which would not be good for the human papillomavirus. So, the high-risk human papillomaviruses have an E6 protein. They produce an E6 protein which is going to stop this from occurring. It's going to put a great big cross for it, so that the P53 pathway will not stop the good work that the E7 protein is doing for the human papillomavirus. So, let's now have a look at the more detailed account of the P53 pathway. So let's have a look at the P53 pathway in a little bit more detail. So we'll start off with the P53 protein itself. So I'll draw a cell here. So let's let this be the cell membrane here. And let's let this be the nucleus here. And let me just do the nucleus again so that it actually makes a complete circle. There we go. Okay, so P53 protein is usually synthesized all the time. So you're always making a little bit of P53. So I'll draw P53 here. So this is going to be the P53 protein. And P53, by the way, is a tumour suppressor protein. It's a very much so anti-cancer protein, and if you get loss of function and mutations in it, that is pro the formation of cancer. So often people call P53 tumour suppressor protein P53. So you're always producing a little bit of P53, and the action of P53 is to go into the nucleus and act as a transcription factor. So I'll just put here, it is a TF, it's a transcription factor. So if it was left to do what it wants to do, it would go from the cytoplasm where it's being produced, because of course the translation machinery is in the cytoplasm, and then it would move back into the nucleus and it would act as a transcription factor, so it would change gene expression. And how is it going to change gene expression? Well, of course, it's going to make things that will stop the cell cycle from occurring, and these have a special name, so I'll just put them here. These are called cell cycle inhibitors. So one of the things that it increases the expression of are cell cycle inhibitors, which are special proteins that can stop the cell cycle. We won't go into the examples of cell cycle inhibitors, and we certainly won't go into the mechanism because that's a whole video in itself, but know that P53 activates the expression of proteins that will stop the cell dividing. That's overall enough for us to know. In addition, if it's activated to a big enough extent, you'll get a lot of pro-apoptotic proteins being produced. So other proteins that it will produce are proteins that are pro-apoptotic, that activate the intrinsic pathway of apoptosis. So pro-apoptotic proteins will also have their expression increased by P53, and if it's activated to a great enough extent, you'll get a lot of these being produced, and then the cell will commit apoptosis. And of course, finally, the other proteins that are going to have their expression increased are DNA repair proteins, so proteins that are involved in repairing damaged DNA. So DNA repair proteins are also going to have their expression increased. So overall, this is what P53 would do. It would be produced, and it's produced continuously. It would go into the nucleus, and it would activate cell cycle inhibitors and pro-apoptotic proteins and DNA repair proteins. Of course, it's not usually allowed to do this, otherwise all cells would be not dividing, committing apoptosis, and activating DNA repair mechanisms. So this is not normally what happens. Something stops the P53 from going back into the nucleus and changing gene expression normally. So we have to add another little protein on here. So I think we'll have this next protein in blue here. So there is another protein that's very important here, which we'll have here in blue, which is called MDM2. Now, MDM2 normally goes along and binds to P53 and prevents it going back into the nucleus and changing gene expression. In addition to just binding to it and stopping it from performing its function, it also acts as a ubiquitin ligase. So MDM2 is something called a ubiquitin ligase, and this is an enzyme which attaches ubiquitin molecules onto the sides of other proteins. And ubiquitin molecules is the normal way in which a protein is targeted for proteasomal degradation. So coming back down here, I talked earlier about proteasomes inside the cytoplasm of cells, and we were talking about it because remember, E7 somehow leads to the retinoblastoma protein being destroyed by proteasomes. And remember, the mechanism of that isn't well understood. The normal way in which proteins are targeted to go into proteasomes is by having ubiquitin molecules attached onto them. So they get ubiquitin molecules attached onto them, and this then is the thing that attaches to the opening of the proteasome. The ubiquitin attaches to the proteasome, and then the protein gets pulled into the proteasome and broken into smithereens. So this is an important molecule to know about, ubiquitin. So drawing a little picture here, and let's have our P53 here in purple. Okay, so this is P53. And then we have MDM2, which has come and bound to it. So I'll put this on here. This is the MDM2. And now, not only does it bind to it and stop it from actually performing its function now, but it's also going to lead to the destruction of the P53. So what you get is the MDM2 sticks ubiquitin molecules, and I'll show this as this square here. So this is representing the ubiquitin molecule. It sticks ubiquitin molecules onto the P53, and these are the things that then tell the proteasome to destroy this P53. This is effectively the label to be destroyed by a proteasome. Ubiquitin is the label to be destroyed by the proteasome. Okay, so we describe MDM2 as a ubiquitin ligase because it is a protein which attaches ubiquitin molecules onto other proteins. So ligate means to attach something onto something else. So this is ligating ubiquitin onto another protein. So overall, this is normally what happens to poor old P53. It's produced and it does not have a long life. It has a very, very sad and short life. It meets this horrible bully called MDM2, which binds to it, stops it from doing what it wants to do, which is go into the nucleus and activate the expression of these genes. And then this MDM2 attaches these horrible molecules called ubiquitin onto it, and it then gets destroyed by the proteasome and cut into small pieces. So it does not have a nice life normally, P53. So how then does aberrant cell division and DNA damage lead to the activation of the P53 pathway? This is why the P53 pathway is usually off. Now let's talk about how you activate it. So if the cell is dividing in a really strange way, or if you've had DNA damage, then intracellular pathways are activated, which are going to do things to P53. So let me just show this. We'll get another colour. So these processes, aberrant cell division and DNA damage, they lead to the phosphorylation of P53, and again I will not be going into the details of the signaling pathways downstream of aberrant cell division and DNA damage which actually lead to this. It's enough for us just to draw an arrow. We can't discuss everything, otherwise we'd be here forever. Okay, so what happens then is these things happening leads to intracellular signaling pathways being activated, which causes the phosphorylation of P53. So P53 gets phosphorylated, and then once P53 has got a phosphate group attached to it, then the MDM2 cannot bind to it, and therefore it survives. It doesn't get the MDM2 binding to it, it doesn't get ubiquitin attached to it, and it can then go into the nucleus and perform its role. So to summarise the P53 pathway, normally P53 pathway is off, and the reason is that when we produce P53 it doesn't last long. It gets bound to by MDM2 very quickly. MDM2 stops it from going into the nucleus, and then this horrible MDM2 attaches ubiquitin onto it, and that causes the P53 to be destroyed by the proteasome. When we want the pathway to be activated, so if the cell is dividing in a stupid way, or if there's been DNA damage, then pathways which we're not going into the details of are activated that result in the phosphorylation of P53, and these phosphate groups stop the P53 molecules from being able to be bound by the MDM2 proteins. So P53 therefore survives, it can go into the nucleus, and it can activate the gene expression of cell cycle inhibitors, pro-apoptotic proteins, and DNA repair proteins, which will then sort out this horrible problem that we've got. So this is an absolutely fantastic pathway. This is protecting the multicellular organism from dangerous cells that might cause problems for the multicellular organisms, that might destroy the multicellular organisms. So this is a fantastic pathway that is inside of all nucleated cells. Of course, red blood cells will not have this pathway, so all nucleated cells have this pathway. Now, this pathway that we've now discussed in a lot of detail, as I've previously said, is a big problem for our high-risk HPVs, particularly the high-risk HPVs, because they particularly cause aberrant cell division. The low-risk HPVs, they have a lower power E7, and therefore they're not as likely to activate the P53 pathway as the high-risk HPVs. So the high-risk HPVs need to have a way of stopping this from occurring, and this is what the E6 protein of high-risk HPVs does. So E6 is basically like a more powerful version of MDM2 effectively. So let's draw E6 again in the evil red over here. So in these proteins in the mid-zone there of the squamous stratified epithelium that are infected with high-risk HPVs, you produce this E6 protein, and the E6 protein binds fantastically well to P53. it binds to P53 even quicker than MDM2 would bind to it. So remember, P53 is continuously being produced, and normally quite quickly MDM2 binds to it and then destroys it. E6 is much better at doing it than MDM2. It binds ridiculously quickly to the P53, and then it is not a ubiquitin ligase itself, but it recruits another ubiquitin ligase that is in the host cell. So I'm just going to draw this one in. We'll have this in yellow here. So inside all human cells, or at least, inside all human cells, you have this protein called E6 associated protein. So here it is, E6AP. So let me just write its name over here. So E6AP is an enzyme. This is a normal human enzyme, and it's a ubiquitin ligase. It is responsible for attaching ubiquitin onto certain proteins. Now, normally, it does not have any role in attaching ubiquitin molecules onto P53. So this is E6 associated protein, E6AP. So this is a ubiquitin ligase, so it's another one of these enzymes, but normally, unlike MDM2, it has no role in usually attaching ubiquitin onto P53, so this does not normally attach ubiquitin onto P53, that's MDM2's job. It attaches ubiquitin onto a whole set of different proteins that aren't relevant to our story. However, this E6 protein that the virus has produced can cause this protein to bind onto this complex. So let's draw this here now. So here comes the E6 associated protein enzyme. It attaches on, it's recruited by the E6 protein there. You can see that quite clearly on my picture. It has been recruited by the E6 protein, and now it can attach ubiquitin onto P53. It does not normally do that. In a cell where you do not have E6, it doesn't do anything to P53. It has no role in attaching ubiquitin onto P53, but E6 can recruit this protein that is a ubiquitin ligase for other proteins to attach ubiquitin onto P53. So this is what's going to happen. The high-risk human papillomaviruses produce this E6 protein that can bind to P53. It can recruit the E6 associated protein, which is a normal human enzyme, ubiquitin ligase, that does not normally have anything to do with P53, which will then add a ubiquitin group onto the P53, and then of course the P53 will be destroyed by the proteasome. And this happens even quicker than MDM2 would destroy the P53, and it happens so quickly that you don't actually even get a chance to phosphorylate the P53 anymore. P53 levels go down in the cell to levels below what they were when the MDM2 was just responsible for its degradation. P53 has an even shorter life cycle. It lives for even shorter amount of time, and you just don't have the time to phosphorylate the P53 before the E6 has resulted in the degradation of P53. So effectively you destroy the P53 pathway in these cells that have this E6 protein. So I will stress again that this is a function of the E6 protein of the high-risk human papillomaviruses. This is not a function of the E6 protein of all human papillomaviruses. Just the high-risk ones seem to have an E6 that can do this. And it turns off the P53 pathway. Now that you've got aberrant cell division, you can still set off the pathways to try and phosphorylate P53, but it's all been destroyed far too quickly for you to find any to phosphorylate, and therefore you do not get the activation of the P53 pathway, you do not get cell cycle inhibitors being produced, you do not get pro-apoptotic proteins being produced, and you do not get the DNA repair mechanisms activated. So E6 turns off the P53 pathway in this incredible way that we've just discussed. So we have now discussed the function of E6 and E7, these partners in crime. E7 causes the cell to divide very rapidly, and E6 in the high-risk human papillomaviruses stops the P53 pathway becoming activated, which would become activated if you just had E7. So overall, the message from this stage of the life cycle is that we have a lot of these mid-zone cells dividing at a very fast rate, and they're all having nuclei that are absolutely bursting full of viral genomes. So this is the productive phase of the viral life cycle. This is the amplification phase. We are producing loads of new viral genomes. And that completes the early phase of the viral life cycle. In the next video, we'll see the later stage of the viral life cycle, where we'll go up into the superficial layer above the midzone, where you'll now start to get the viral genomes packaged into capsids, and you'll then get the release. We'll also go over transmission and the immune response. Okay, so we'll do that in the next video.